Hello, my name is Susan, and today I'll be walking you through my poster, Sox-based Sensor Phosphopeptides for Continuous, Homogeneous, and Quantitative Monitoring of Protein Phosphatase Activity, that I presented recently at the 2023 AACR meeting in Orlando. Activation of oncogenic signaling pathways by protein phosphatases can drive tumorigenesis and malignant transformation. In addition, some phosphatases increase T cell sensitivity and function. This reinforces the need for protein phosphatase inhibitors and activators for cancer therapy. Two recent publications, one by Sivagenesh et al. And the second by Stanford and Bottini in Nature Reviews in Drug Discovery in 2023, review recent work in directly targeting phosphatases in cancer. They look at reversible competitive inhibitors, an uncompetitive inhibitor, and an irreversible inhibitor that target the active site. They also look at allosteric inhibitors for WIP, SHP2, etc., a protein-protein inhibitor for calcineurin, and a protein degrader for SHP2. Assay Quant utilizes PhosphoSense technology developed by Dr. Barbara Imperiali at MIT to develop assays for phosphatase inhibition or activation. A natural peptide is synthesized containing a phosphorylated serine-threonine or tyrosine residue with a C-SOX moiety. This substrate has inherent fluorescence, which decreases over time when it is dephosphorylated by a phosphatase in the presence of magnesium. This decrease in signal can be monitored kinetically over time, which also allows rates to be determined, or it can be used to calculate a signal to background for an endpoint assay. A second assay that can also be utilized as an endpoint assay is our redshift assay, which measures time-resolved fluorescence. After the standard assay is complete, europium is added to displace magnesium and bind to the peptide. After reading the assay at emission 620, a signal to background can be determined. The advantage of this assay is that it prevents compound interference that may be observed at a lower wavelength. This slide illustrates examples of some assay development. Panel A shows substrate screening for WIP1 phosphatase. A panel of substrates is tested where we look for the optimal dephosphorylation rate. The two best substrates are AQT-O525 and AQT-O523, where we see a strong decrease in rate with the yellow and light blue lines respectively. Panel B shows an enzyme titration for PTP1B. A complete dephosphorylation is observed at 10 nanomolar PTP1B. As you decrease the amount of PTP1B, the curves increase until they reach the control rate without phosphatase, which is the blue line. Since the reactions are run kinetically, by taking the rate at each enzyme concentration, one can easily determine the linear range of the assay. Panel C shows a redshift assay for PTPN2 with AQT-O264. AQT-O427, the unphosphorylated version of AQT-O264, is run as a control. The signal observed with AQT-O264 alone is decreased sevenfold with the addition of 0.25 nanomolar PTPN2, which is shown by the blue bars. The red bars show the unphosphorylated substrate, which are comparable to AQT-O264 with PTPN2, which indicates complete dephosphorylation. The Z prime for this assay is 0.94, which indicates an excellent assay. The following two slides show examples of phosphatases implicated in cancer. Panels A and B show tyrosine phosphatases, and panel C shows a dual phosphatase. In the plots, the black lines show the control, and the yellow lines show the substrate in the presence of phosphatase, which dephosphorylates the substrate. The green lines show the assay run in the presence of sodium orthovanadate, a known tyrosine phosphatase inhibitor, at three concentrations, 1, 10, and 100 micromolar. As observed, inhibition of phosphatase activity with increasing amounts of sodium orthovanadate approaches the control black line. The two tyrosine phosphatases are inhibited better than the dual phosphatase. Panels A and B show calcineurin and PP2A alpha, serine-threonine phosphatases. The green bars only begin to reach the black control line at 100 micromolar sodium orthovanadate, which is expected as sodium orthovanadate is a strong tyrosine phosphatase, but will inhibit some serine-threonine phosphatases at high concentrations. This slide illustrates our phosphatome profiling panel, where we profile an inhibitor against a panel of phosphatases to check for specificity. New phosphatases are always being added, and we can add additional phosphatases of interest upon request. In summary, we have shown a recent renewal of interest in phosphatases as cancer targets and the development of a robust activity-based assay for drug discovery that is ideal for assessing enzyme regulation and elucidating mechanism of action, potency, and specificity through profiling.